Hello everyone, we are iGEM 2015 CUHK team. In this video, we are going to introduce you to the concept of synthetic biology, which is a major focus of iGEM and our project on Magnetobeta violandi. So, synthetic biology, you've probably heard it somewhere before. It is actually a combination of biological disciplines like biotechnology, molecular biology, and systems biology. In many ways, it involves the direct manipulation of an organism's genome. Essentially, it is the design and construction of new biological parts, devices, systems, and redesign of existing natural biological systems for useful purposes. In synthetic biology, the most used standardized DNA parts are called biobricks. They are used to design and assemble synthetic biological circuits, which are then incorporated into living cells to make new biological systems. Examples of biobrick parts include promoters, ribosomal binding sites, and terminators. And the iGEM Foundation is an organization dedicated to the advancement of synthetic biology since 2004 by education, competition, and community projects. The iGEM competition in which our team is taking part has now already expanded to 280 teams, reaching more than 30 countries from all over the world. So, what is our project about? Our project for iGEM 2015 is Magnetobeta violandi, magnetosome-forming acetobeta violandi with downstream applications. The main idea is to produce magnetic nanoparticles in the rapidly growing bacteria Acetobeta violandi. These bacteria are chosen because they can grow in aerobic conditions, but also provide an intracellular and aerobic environment which is required for making nanoparticles. A magnetic nanoparticle consists of magnetite and magnetosome membrane. Magnetite is one of the naturally occurring iron oxides, and magnetosome is a membranous structure surrounded by a lipid bilayer with specific proteins sorted to the membrane. Magnetic nanoparticles are originated from the bacteria called Magnetospirillum grifols-waldens. In this project, we are not just trying to move the magnetotic biogenesis machinery to a foreign organism, but we've also developed two new downstream applications. First, we aim to fuse recognition elements with the transmembrane proteins that are tightly bound on the magnetosome membrane. After binding, we can control the magnetosomes by attracting them with external magnetic force. Then we can wash and elute the product we want. An example is the heavy metal binding peptide, which captures heavy metal ions in polluted water for water treatment. Another application is immunoprecipitation, in which antibodies on the membrane capture proteins of interest for protein extraction and purification. A major application is water treatment. Water pollution is a serious environmental issue that demands our attention. Different kinds of heavy metal ions, like lead and copper, are found in the marine system. To address this issue, we try to fuse elements that recognize specific heavy metal ions with the metal binding peptides on the magnetosome membrane, so the various kinds of heavy metal ions can be attracted by biomagnetic force of the magnetosomes. In this way, heavy metals from polluted water can be removed along with the removal of the magnetic nanoparticles. Second, we would also like to control the movement of acetobeta as a component in a microbial fuel cell, or MFC in short, by external magnetic force. We have designed a self-sufficient system that produces hydrogen by mutated nitrogenase and, at the same time, releases electrons to the anode and recycles the protons for ATP production by membrane-bound hydrogenase. We expect an electricity production at 30 mA within an electrode surface area of 0.83 square meter. And this is actually a twofold increase compared to a stainless steel mesh biocathode MFC. So, this is a general overview of our project from iGEM CUHK Team 2015. Hope you guys enjoy it, and see you!